Welcome back, class 12, to the online bio classes. Sixth chapter, that is molecular basis of inheritance. I will give the summary of the entire chapter. First one is DNA, structure of polynucleotide chain. The structure of polynucleotide chain, DNA stands for deoxyribonucleic acid. DNA and RNA, both of them are the nuclear material. Although RNA was the first nuclear material to be discovered, but DNA is structurally more stable and active than RNA. And DNA is a polymer of deoxyribonucleotide. Now in your book you will get the word DNA is full form, you can learn deoxyribonucleotides. If it is RNA, it stands for deoxyribonucleic acid, you can write, or nucleotides. So DNA is, the full form of DNA is deoxyribonucleic acid. Don't write nucleotides, nucleic acid you write. Nucleic acid. Okay. So D means deoxyribonucleic, nucleic and acid. Now similarly we have the RNA, that stands for ribonucleic acid. So both of them are nucleic acids, the material which is present inside the DNA or in the nucleus of a cell. Now nucleoside, what is nucleoside? It is asked. It is nitrogenous base. The word is nucleoside. If asked what is nucleoside, so how can you learn? It is equal to nitrogenous base. Nitrogenous base. You know there are two types of nitrogenous base is there, purines and pyrimidines, that is adenine, thymine, cytosine, guanine. And plus it consists of the pentose sugar, means the sugar which has 5 carbon atoms in it, through N-glycosidic bond, means both of them are attached with each other with the help of a bond, and that bond is called N-glycosidic bond. Glycosidic bond. Okay, so what is the function of this glycosidic bond? It helps to attach the pentose sugar, that is the sugar molecule with the 5 carbon atoms with the nitrogenous base. Okay, attachment is done with the help of the bond which is called glycosidic bond. Now what is nucleotide? Let's come to the point. Next one is nucleotide. Nucleotide is equal to, means the meaning of nucleotide is nucleoside plus the phosphate group. This one already we wrote. Nucleoside plus the phosphate group, that is PO4, you know, formula of phosphate. So phosphate group. Phosphate means PO4. Alright? When both of them are present together, it is called a nucleotide. And just now we said that DNA is a very long polymer of nucleotides, means many nucleotide units are attached to each other to form a long chain, that is called DNA. Now, what is the name of the bond which is present between them, between the nucleoside and the phosphate group? The bond which is present, that is called phosphodiester bond. So remember the name also, phosphodiester bond. If asked where is this phosphodiester bond found, it is present between the nucleoside, ER not written, and the phosphate group. That makes a nucleotide. Clear? Now next one is, many nucleotides link together through that 3-5' phosphodiester bond to form a polynucleotide chain, as it is present in DNA and RNA. In the course of formation of a chain, a phosphate moiety is set at 5' end of the ribose sugar, that is called the 5' end of polymer chain, and one OH group, that is hydroxyl group, remains free at the 3' end of the ribose. Okay, now double helix model of structure of DNA. This one you have already studied. Scientists who are involved, I will say the names and their contributions in double helix model of structure of DNA. Number 1, Friedrich Miescher, M-E-I-S-C-H-E-R. What he discovered? He first identified DNA as acidic substance present in the nucleus, and he called it as nuclein. So Miescher, M-E-I-S-C-H-E-R, this scientist discovered the nuclear material inside the cell and called it as nuclein. And Wilkins and Franklin, the second scientist, Wilkins and Franklin, both of them produced X-ray diffraction data for the structure of DNA. Third one is Watson and Crick. Both of them, this Watson and Crick, they proposed the double helical model of DNA, that is the structural model for DNA, and this model was based on the X-ray diffraction data which was obtained by Wilkins and Franklin. So it is there in your syllabus. What is the contribution of Wilkins and Franklin? Wilkins and Franklin, both of them produced X-ray diffraction data for DNA structure. So if you want, I will write it so that you can understand the contribution of different scientists. It will be short and easy to learn. Okay, scientists, like first one I wrote, Friedrich Miescher, spelling Friedrich Miescher. Okay, M-A-I-S-C-H-E-R. First identified DNA as an acidic substance. First identified DNA as an acidic substance. Okay, present in the nucleus and named it nuclein. Nucleus and named it nuclein. This word nuclein was given by Friedrich Miescher. Okay, his contribution that he was the first to identify DNA is an acidic substance which is present inside the nucleus of a cell, and he didn't call it as DNA. He said that the word nuclein he gave the term. Second one is Wilkins and Franklin's. Wilkins and Franklin's, they produced Wilkins and Franklin. These two scientists, what they did, they produced X-ray diffraction data for DNA. Produced X-ray diffraction data for DNA structure. X-ray diffraction data for DNA structure. Okay, the next one is Watson and Crick. They proposed double helix model for the structure of DNA. Watson and Crick proposed double helical model of DNA. Double helix or helical, any word you can use, double helical model of DNA. And their proposition was based on X-ray diffraction technique or the data. Then last one is Erwin Chargaff. Chargaff's rule it is called. Erwin Chargaff, C-H-A-R-G-A-F-F. Erwin Chargaff, he was the one who gave the nitrogenous base pairing rule. So he proposed that in double-stranded DNA, ratio of A by T and C by G remains the same. That is A by T when C by G is equal to 1. The ratio of A is to T, that is A by T upon C by T, C by G, not T, is equal to 1. So it means A by T is equal to 1. That is same. It remains same and they are equal to 1 only. So A by T is equal to C by G is equal to 1 only. It remains unity. Okay? It means they propose that the ratio of A is to T by C is to G, they remains 1. Now next is, what are the features of double helical structure of DNA? The features of double helical structure of DNA that we have to study. Okay, first one is in a DNA, both the polynucleotide chains, they are coiled to form a helix. Like as you can see in the diagram, this one is a polynucleotide chain. They are coiled to form a helix-like structure. Okay. Similarly, this is also one of the strand of DNA. Okay. Now, two polynucleotide chains are there. What are they? What do they represent? They represent polynucleotide chain. Poly means long, nucleotide means many units of nucleotides are there, chain. So what is DNA? DNA is a long polymer of polynucleotide chain. It is coiled to form a helix. As you can see, the coiling is there. It is twisted or coiled to form a helix, just like a spiral staircase is there. Sugar phosphate are present in the backbone, that is, they form the structure of this helix. And the complementary base pairs, they are present in between these two strands. Like here, if A is there, then here it will be adenosine, thymine, cytosine, guanine. They are bonded with double bond and here with triple bond. Okay, so what is the bond pairing? Complementary base pair means adenine is complementary to thymine and cytosine is complementary to guanine, and they are attached with the help of this bond is called hydrogen bond. Okay, so complementary base pair with each other through hydrogen bonds. Purine always pairs with corresponding pyrimidines, like adenine pairs with thymine with two hydrogen bonds, as I have shown in the diagram. If it is not clear, like this A double bond T, this is adenine, this is thymine. Cytosine pairs with guanine with a triple bond. So this one shows the triple hydrogen bond and this one shows the double hydrogen bond. One is purine, the other one is pyrimidine. Okay, they form complementary base pairs. Now, the adenine pairs with thymine and guanine pairs with cytosine with 2 and 3 hydrogen bonds respectively. Now, the helix is right-handed, as you can see, right-handed, and the pitch is 3.4 nm, that is angstrom. Like if one of the chain is 5 dash, it will terminate in 3 dash. If this one starts with 3 dash, it will terminate or end in 5 dash. Okay, the ends. And 10 base pairs are there in each turn, like from here to here, one turn will be there. Then in one turn, the total number of base pairs which are present, it is equal to 10 base pairs. And the plane of one base pair stack over the other in a double helix. This proves the stability of the helix along with the hydrogen bonding. Now next one is packaging of DNA helix. You know that the distance between two base pairs of DNA is 0.3 nm. Like in between these two base pairs, the distance is only 0.3 nm. That is equal to 3.4 angstrom. Already we had done earlier. And the total number of base pairs in human beings is 6.6 into 10 to the power minus 9 base pairs. So 6.6 into 10 to the power, not minus, 9 BP. That is base pairs. This is the total number of base pairs in the human DNA. So total length of human DNA, it is almost equal to 2.2 meter. This much long, it is in meter. So such a long polymer of DNA, how can it fit into a small nucleus which is not visible or microscopic? So 2.2 is too large to be accommodated or packed inside the nucleus, whose size is 10 to the power minus 6 meter. This is size of the DNA, 2.2 meter, and this one is in minus 6. This is the size of the nucleus. So nucleus having such a small size in minus, that is 1 by 10, 6 zeros will be there. In such a small place, how can such a long DNA polymer be fitted or accommodated? So that we have to study in packaging of DNA helix. Okay. So here we find that organization of DNA in case of prokaryotes and eukaryotes, both of them are different. Prokaryotes means those living organisms in which the nuclear material is not present inside a nucleus, but it is dispersed in the whole cytoplasm, like bacterial cells are prokaryotes. They are the primitive cells. So in case of prokaryotes, they do not have the nucleus, and the DNA is fully scattered in the full cytoplasm. Like this DNA, this nucleus is only not there. So DNA is present everywhere in the cytoplasm, scattered in the whole cytoplasm. In certain regions called nucleoids, DNA, actually what is the charge of DNA? If it is asked, you will write DNA is a negatively charged particle. Means negative charge is here. Okay? So DNA being negatively charged is organized in large loops and held by the proteins. The proteins are positively charged there. So that is forming a structure which is called nucleosomes. Okay? Now next, in case of eukaryotes, so what we found? That in prokaryotes, DNA is scattered in the full cytoplasm because there is no nucleus, and in certain regions called nucleoids, DNA which is negatively charged is organized in big loops which is held by some proteins which are positively charged. So we find that DNA is negatively charged and it is held by some positively charged proteins which are present, okay, in case of prokaryotes. Now when we talk about the eukaryotes, that is all the modern cells with a well-defined nucleus and the nuclear material, we find that they have a positively charged basic protein which is known as histone protein. You have already studied. Histone proteins are positively charged. They are the basic proteins. Why they are positively charged? Because they contain the basic amino acid residues like lysine and arginine. Due to the presence of basic amino acid residues like lysine and arginine, they are positively charged. Okay. Now histone octamer. Histone octamer means 8 molecules of histone are coiled together to form a structure called histone octamer. 1, 2, 3, 4, 5, 6, 7, 8. So they form, these all are proteins only, histone proteins. So when they are present together, the unit of 8 molecules of histone, it is called histone octamer. Okay, I will just rub it. So what is histone octamer? A unit of 8 histone proteins is called histone. Octa means 8. Now next one is DNA which is negatively charged. It is wind means it is wrapped around the histone octamer to form a nucleosome. In your book also you can see one diagram is there like this. This one shows F1 histone, H1 histone is written. H1 histone, it is the name of the. So this is one of them. Now you will find that the core of histone molecules are there, means this one a circle is made in which the histone molecules are there, and around these histone molecules the DNA is wrapped like this. Okay, this is the DNA which is wrapped. So this is structure in which the negatively charged DNA is wrapped around the positively charged histone octamer. It forms the nucleosome. Okay, got it? So nucleosome is made up of a core of histone molecules which is wrapped around by the DNA that is negatively charged. Okay, so you can see one more coiling has been shown like this in the diagram. Now next is, one nucleosome has how many base pairs? It has approximately 200 base pairs of DNA. Means in a DNA, the total number of base pairs are there. One particular nucleosome contains this much base pairs. Then nucleosome in a chromatin, it resembles just like beads on a string. Like if you take a string and on the surface of a string certain beads or swellings are there, like them only these nucleosomes resemble. So what are they? They are the nucleosomes. And this string represents the chromatin. This represents the, okay, that one is called the, these beaded particles which are present on the string-like structure, they are called the nucleosomes, and beads on this string structure in a chromatin, they are further packaged to form the chromatin fibers. Like you know that inside the cell the nucleus is present. The nucleus contains thread-like structures which are called chromatin network. So this chromatin network will condense together and it will form the chromatin fibers which will coil and condense to form the thick chromosomes, and the chromosomes are the one which contains the hereditary information. The units called genes are present in it. Okay, so during metaphase, you can find that they are arranged in the center of the nucleus. Okay, these chromosomes. Now, next we have to study is there are certain non-histone chromosomal proteins, NHC, non-histone chromosomal proteins. NHC, full form is asked in the examination. What is the full form of NHC? It stands for non-histone chromosomal. They are extra, okay, non-histone chromosomal proteins. So NHC stands for non-histone, means they are not histone protein, they are some other protein. Non-histone chromosomal proteins. They are the additional set of proteins which is required to pack the chromatin at higher level. So you understood now how that large amount of DNA is being packed inside a small nucleus of the eukaryotic cell. Got it? You got the idea of what is nucleoside, how it is different from nucleotide, what is octamer, histone octamer. It is made up of 8 molecules of histone. What is the charge on DNA? It is negatively charged. What is the charge on histone? It is positively charged. What is the difference between prokaryotes and eukaryotes in terms of DNA packaging? In terms of prokaryotes, the DNA material is fully dispersed in the whole cytoplasm because there is no nuclear membrane, and it forms certain structure called nucleoid that binds with the proteins. And in case of eukaryotes, you found that there are the histone molecules which forms the nucleosomes. These histone molecules forms 8 histone molecules form octamer, histone octamer, around which the negatively charged DNA wraps around to form a structure called nucleosome, and the nucleosome appears like beads on a string which are present on a chromatin, and this chromatin network condenses during the cell division to form the thick chromosomes which are the site of, or which are the place of genetic information. Okay, so now next we have to study discovery of DNA as a genetic material. We found that through the principles of inheritance and discovery of chromosomes in nucleus, it was done long time back, but still there was a confusion whether the protein is the genetic material or the DNA is the genetic material. So in order to prove which one is the genetic material, a scientist called Griffith, he performed an experiment on the bacteria called Streptococcus pneumonia, the one which causes pneumonia disease. It has 2 strains. The Streptococcus bacteria is of 2 types. One is R strain, that is rough strain of bacteria. The other one is S strain, that is smooth strain. So R strain is the one which is non-virulent, that do not cause the disease. Okay, R strain and S strain, 2 types of bacteria are there. The R strain bacteria produce rough colonies on the culture plate, okay, whereas they do not have any polysaccharide coat in R strain, but in smooth or S strain bacteria, they produce smooth colonies. So what Griffith had done, I will say in short, Griffith performed experiment with the bacteria called Streptococcus pneumoniae, and he found that when the live R strain, that is the living R strain bacteria containing injection was injected into a mice, the mice died. Why? Because it was the virulent type, the R strain bacteria, non-virulent type. So when living virulent strain, that is S strain, was injected, when S strain, this is the virulent one, the one which causes the disease. This one is the non-virulent. When S strain was injected into the mice dies. Okay. Then second one, when living a virulent strain was injected, that is when R strain was injected into a mice, the mice is alive, not dying. First he injected the S strain, that is the virulent type of bacteria, in the mice, and the mice dies. Then he injected the bacteria, that is R strain bacteria, which is non-virulent type of bacteria. Then the mice was alive. In the third case, what he did, he heat-killed virulent type, that is S strain, which is killed by heat. The killed S strain was injected in the mice. then what happens? The mice will remain alive, it will not die. Why? Because already the bacteria is killed by heat. But in the fourth and the last step, what he had done, he took the living avirulent strain, that is the S strain, plus this one also, the heat killed S strain, heat killed virulent type and living avirulent type. Living avirulent type means avirulent one is the R strain, sorry. So he took the R strain and the heat killed S strain. So this one you can take here and then injected into the mice and the mice dies. Now you can see earlier the R strain was avirulent, not causing the disease. At last you find that when the R strain combines with the heat killed S strain bacteria, the mice will die. It means something has changed the DNA of that R strain bacteria, which was earlier avirulent, into virulent type, and this was possible only because of DNA. Understood? It means when heat killed strain was there, bacteria, heat killed or the S strain bacteria were there, still their DNA was active and it helped in the transformation of R strain DNA into the S strain DNA. So live R strain in the presence of S strain, that is heat killed S strain, they produce the disease because somehow the R strain bacteria is transformed by this heat killed S strain bacteria. Means the DNA is copied or it is transferred. So you can say that it was concluded that there must be some transfer of genetic material. Okay, and this biochemical nature of transforming material, it was proved by three scientists like Avery, MacLeod, McCarthy. They worked to determine the biochemical nature of genetic material which was responsible for transformation, and through their experiment they found that DNA has to be the genetic material. The next one was Hershey and Chase experiment, which was done on bacteriophage. Bacteriophage is the name of the virus which causes infections to bacteria or that infect bacteria. So when a bacteriophage infects bacteria, the viral genetic material gets attached with the bacterial genetic material, and bacteria then treats the viral genetic material as its own and starts synthesizing more of the viral particles. So Hershey and Chase worked to discover whether it was a protein or a DNA that entered the bacteria from the virus. So they used the radioactive sulfur and phosphorus for their experiment, and they found that when they infected the E. coli bacteria with the radioactive phages, that is the virus containing radioactive material inside their body, E. coli was blended, means mixed, and centrifuged to remove the bacterial DNA, to remove the viral particles. It was seen that the bacteria with radioactive DNA were radioactive, while those with radioactive proteins, they had lost their radioactivity. It means proteins is not the genetic material which was transformed from one cell to the other. So this shows that it is DNA which enters the bacteria from viruses and not the proteins. So through this experiment, Hershey and Chase proved DNA is the genetic material. Okay. Then what are the properties of genetic material? Number one, it should be able to replicate. Replicate means to make a copy. Like one copy of DNA is there, it should be able to make its copy, reproduce, we can say in simple words. Okay. Then duplicate and produce identical copy. It should be chemically and structurally stable. Like DNA is more stable than RNA. Number three, it should have a scope for changes so that evolution can take place. We know that how living organisms evolution is caused due to changes in the genes or DNA, which brings about appearance of new characters and help them to survive in the changing environment. And it should follow the Mendelian principles of inheritance, that is Mendelian law of inheritance, like law of dominance, law of segregation, independent assortment. Then we studied about the differences between DNA and RNA. DNA has deoxyribose sugar, RNA has ribose sugar. In DNA, DNA is mostly acting as a genetic material in most of the organisms, but RNA acts as a messenger and adapter. It also acts as a genetic material, but only in some viruses. In DNA, uracil is present, but in RNA, 5-methyl uracil is absent. Uracil is not present in DNA. In RNA, uracil is present in place of thymine. In DNA, we have the thymine base, whereas in RNA, it is replaced by uracil. Then DNA is stable, whereas RNA contains 2 hydroxyl group, which makes it biodegradable, and so it is not stable. Chemically, DNA is less reactive and it will undergo mutation very slowly, but RNA mutation is very fast. DNA requires RNA for protein synthesis, that is, from DNA, messages are copied to RNA, and then from RNA the protein is synthesized. This is also called the central dogma. Okay, how the information passes from DNA to RNA, RNA to proteins. So last one, RNA. In case of DNA, DNA requires RNA to make proteins, but RNA itself directly can make proteins with the help of ribosomes. Now, why DNA is more stable than RNA? If question is asked, the answer is, in RNA, 2 OH group is present at every nucleotide. OH means hydroxyl group is present. At every nucleotide, this OH group is present, and this makes RNA unstable and degradable. Then second reason why DNA is more stable is DNA contains thymine in place of uracil, which gives extra stability to DNA. And the last one, RNA is a biocatalyst, so it is more reactive. And one more point, DNA is double stranded, making it more stable and resist the changes by the method of repair mechanism. Now, what is DNA replication? DNA replication is the phenomena or process by which a duplicate copy of DNA is synthesized. In replication, the 2 strands of DNA helix, like we had drawn this one, this way I had drawn, no? So they will separate from each other. Both of them will unwind. Okay? And now each strand acts like a template. Template means they will act like a ground level, and then they will synthesize their own complementary strand will be synthesized. Okay? So a new strand is synthesized. Okay? After completion of replication, the 2 copies will have one parental and one newly synthesized strand. Like this is the parental strand that comes from the parent, you can see, from the old DNA. And this one is the new strand which is being produced, synthesized. So one parental strand is there and one newly synthesized strand is there. This scheme of replication, in which even the parental characters are also preserved, is called semi-conservative mode of DNA replication. Then we have experiments to prove that DNA replicates semi-conservatively, which was performed by Michelson and Stell, in which they had grown E. coli in a medium containing nitrogen 15 as the nitrogen source. Another one, that nitrogen 15 was incorporated, that is, it entered into the newly synthesized DNA, as well as the DNA became heavy DNA. Heavy DNA molecules can be differentiated from the normal DNA by gradient centrifugation using cesium chloride as a gradient. Then the cells were again transferred in another medium with nitrogen 14 as the nitrogen source. Then samples were taken randomly and their DNA was extracted. E. coli replicates after every 20 minutes, means in 20 minutes one E. coli bacteria will become 2, 2 will become 4, 4 will become 8. So after 20 minutes, 40 minutes, we take the samples and study them. We found that DNA which was extracted after 20 minutes had a hybrid density, means it had the nitrogen 15 and 14, both of them present in it. Whereas when the DNA sample, or when the DNA was extracted from E. coli after second generation, that is after 40 minutes, it was found that it had equal amount of hybrid and the light intensities. So next we have to study mechanism of DNA replication. Okay. Okay, so next class we can continue with the mechanism of DNA replication, and you can continue with this chapter. Write the notes, or you can write the question answers like Griffith's experiment, and you can also write this question. Experiment, what is the, explain Michelson and Stahl's experiment with the help of a diagram. What is DNA replication? Then next question you can write differences between DNA and RNA, tabular difference you can write. Next, what are the properties of genetic material? Explain Hershey and Chase experiment with the help of a diagram. Then differentiate between R strain and S strain bacteria. Then you will write what is the difference in the organization of DNA in prokaryotes and eukaryotes. Only this much you can do as homework. Thank you.